Hello, in this video I want to talk about the differences between gray matter and white matter. In order to understand these differences, we need to review the structure of a typical neuron. So neurons are the main cells of our nervous tissue, and a neuron has three main parts to it. It has a cell body that contains the nucleus and most of the other organelles, and branching off the cell body we find dendrites and an axon. Dendrites are typically shorter branches, and their job is to bring a signal into the neuron. And the axon is typically a single long branch that's sending a signal away from that neuron. When we look at gray matter versus white matter, we will see different parts of neurons in these different types of matter. Gray matter is mostly composed of cell bodies and dendrites of neurons, whereas white matter is mostly composed of axons of neurons. Gray matter is gray in color because the cell bodies are dense. They have a lot of stuff inside of them, as I mentioned, a nucleus and organelles, and that density makes them appear darker. The axons, on the other hand, tend to be myelinated. Myelin is a fatty layer that covers the axons, insulating the axon and allowing an action potential to travel really quickly down the axon. Now, because this myelin layer is made of fat, fat often appears as light in color, and so that's where we get the white matter from. These myelinated axons are white in color. If you look at the histological slide on this picture here, everything is stained purple, right? But if you look at the two different sides, we have gray matter on one side and white matter on the other. If you look at the gray matter side, you'll notice that we have way more cell bodies of neurons there. So those darker purple splotches are the cell bodies. So there's lots of cell bodies and dendrites in gray matter. If you look at the white matter side, we don't see so many cell bodies there. We mostly see these little white patches here and there, and those are cross sections through myelinated axons. So the axons tend to be found in the white matter, whereas cell bodies and dendrites are found in the gray matter. In the bottom picture, we can actually see a human brain, and a small section has been removed from the brain, which allows us to see the white matter towards the center and the gray matter towards the outside. I want to talk a little bit more about axons and white matter. Okay? So because white matter is composed of axons, the function of white matter is the same as the function of axons. Axons have the job of carrying action potentials, so the job of white matter is to carry an action potential. Now, when we look at axons in the nervous system, right, we can see bundles of axons in the brain and spinal cord, in the central nervous system, in other words, but we can also see bundles of axons in the peripheral nervous system. And we have different names for these bundles of axons. A bundle of axons in the central nervous system is referred to as a tract. A bundle of axons in the peripheral nervous system is referred to as a nerve. Let's look at an example. So your eyeball has attached to it the optic nerve, and the job of the optic nerve is to carry action potentials away from your eyeball towards your brain. Now, the optic nerve is called a nerve when it's in the peripheral nervous system. So at a structure called the optic chiasma, the right and left optic nerves cross over and then enter into the brain. So these are the same axons that were coming off the eyeball and that are now going into the brain. But because the axons are now in the brain, we no longer call it the optic nerve. It's now called the optic tract. It's the exact same axons in the nerve and the tract, but we give it a new name when it's in the brain. Kind of like Lake Michigan and Lake Huron, right? They're both connected, right? They're connected at the Straits of Mackinac, but the water is flowing through both Lake Huron and Lake Michigan, right? We just give it a new name when we cross over from one location to another. In the other picture, we can actually see a cross-section through the brainstem, and attached to the brainstem, we have many cranial nerves. The cranial nerves are shown in white in this picture, and they are part of the peripheral nervous system. So take, for example, cranial nerve number 3 up towards the top. That's the oculomotor nerve. This is a nerve that's carrying action potentials towards the musculature of your eye. But if you look actually in the brainstem, the pink area labeled number 3, that is the oculomotor tract. So it's called a tract because now we're in the central nervous system. So gray matter is made of cell bodies and dendrites. The job of a dendrite is just to bring a signal into a cell body ultimately. And the job of the cell body is really important because it has to sum up all of this incoming information and then decide what to do about it. So cell bodies essentially are processing information. Because gray matter is made of cell bodies, the function of gray matter is to process information. Now, when we look at groups of cell bodies in the central nervous system versus peripheral nervous system, we give different names to those groups of cell bodies, just like we did for bundles of axons. We had tracts and nerves for bundles of axons. With cell bodies, we're going to have ganglia and nuclei. A group of neuron cell bodies in the peripheral nervous system is called a ganglion, ganglia for plural. A group of neuron cell bodies in the central nervous system will be called a nucleus, or nuclei for plural. Let's take a look at some examples. On the bottom of the screen, we see that same picture of a cross-section through a brainstem that we saw on the previous slide. But I'd like to draw your attention to the nuclei shown in the picture. So we already looked at the nerve versus tract in this picture. But what I didn't show you is that at the end of each tract, sort of like an enlarged pink area, we have something called a nucleus. So the arrows here are pointing to the nuclei of these cranial nerve tracts. So at these nuclei, we're going to find groups of neuron cell bodies. And essentially, these are going to be processing the information coming in from that nerve and tract or going out to that nerve and tract. Now let's take a look at an example of a ganglion. We're going to use the example of an autonomic ganglion. So don't forget, a ganglion is just a group of neuron cell bodies in the peripheral nervous system. Okay. So up towards the top of this picture here, we see a brain and a spinal cord and a heart and something weird in between, right? So that weird stuff in between is where we're going to concentrate. That's the autonomic ganglion. Okay, so notice that we're not in the central nervous system. We're outside the brain and spinal cord. So we're in the peripheral nervous system. And if we're going to have a group of neuron cell bodies in the peripheral nervous system, it's called a ganglion. So autonomic ganglia are really important for helping to send signals out towards your visceral organs for both sympathetic and parasympathetic input. So speeding up the heart, slowing it down, that kind of thing. And a ganglion is just an area where we're going to have a group of neuron cell bodies that are going to be receiving signals from another neuron. So in the magnified picture, we can see a cell body inside the ganglion. Now, I'm only showing one cell body inside this ganglion for simplicity's sake, but in reality, there would be multiple cell bodies in this ganglion, and they would be processing information coming from the central nervous system so that this cell body in the ganglion can make a decision and then possibly send a signal out towards the visceral organ.